Good morning and thank you. First, I want to start by thanking Martin and the organizers for the invitation to come and participate in this meeting. As you can tell from the title of my talk, we'll be talking about enteric viruses in poultry. So for our time together this morning, first I'd like to just give a short introduction to enteric diseases of poultry. Then I'll talk about the pathogenesis of enteric viral disease, talk about some of the enteric viruses that we're most interested as they relate to disease syndromes in the field, challenges that we've experienced in diagnosing these diseases, and then just to illustrate an example of some applied research that we've done with my group at UGA, how we identified an etiology for running and stunting syndrome. And then I'll end by just sharing with you some of the recent advances that we've been fortunate enough to experience in the field of enteric viruses. So this is probably not anything new to this group. We've been looking at the avian digestive tract for a day now. But there are a couple of aspects of the GI tract of poultry that I think are extremely important, especially as we start to look at viral diseases. First, we know that the rapid growth rate that occurs during the first week of age is tremendous. The GI tract grows at a rate 5 times greater than that of the rest of the body just during this first week. The duodenum is the primary site of nutrient digestion and absorption, and so any impairment to the GI tract can certainly affect the intake of these nutrients during this critical growth time in the chick. In the small intestine, the villus length doubles in the first 2 weeks of life, and this enables these growing birds to digest increasingly a variety of feed ingredients. And the GI tract doesn't actually reach maturity until 2 to 3 weeks of age. So when we think about diseases of the GI tract, one of the common outcomes is the appearance of undigested feed in the feces. It's a common indicator that something is not going well in the bird. So enteric diseases, as we've learned over the past day, are difficult to manage, and we have a lot of limitations in our diagnostic procedures, but also we have many contributing factors. We have genetics that can contribute to the disease disposition of birds, nutrition, management, the environment, and of course, infectious diseases. We've heard a lot about the bacteria and their contributions to disease and management. We'll hear more about the contributions or role of protozoa this afternoon, but as a clinical avian virologist, we'll talk this morning about viruses that play a role in some of these diseases. As you'll see from this laundry list here of enteric viral diseases, the identification is limited to clinical descriptives. So we have a lot of syndromes listed here rather than specific etiology. In turkeys, we have poult enteritis and mortality syndrome, or PEMS. We have turkey viral enteritis, spiking mortality of turkeys, and poult enteritis complex. All very complicated polymicrobial diseases in turkeys. In chickens, infectious stunting syndrome, malabsorption syndrome, pale bird syndrome, and runting and stunting syndrome. There are some common features that all of these syndromes share. One of the primary features is that most of our enteric viral infections actually occur during the first couple of weeks of age. So there's definitely an age-related susceptibility. The younger the bird, the more susceptible they are to clinical disease. Many of these diseases share similar clinical signs and gross lesions. So it's very hard to look at a clinical presentation in the field and know for certainty that a particular virus is the causative agent. Common signs are diarrhea and depression. We see lack of uniformity, and again, very difficult to associate a specific virus to what we see in the field. This is a very economically important group of viruses, since the GI tract is responsible for uptake of nutrients and conversion to protein and meat. Any assault on the GI tract can result in permanent damage, causing a delay in growth. We might see some mortality or the need to cull before birds go to the processing plants, and certainly an increased susceptibility to secondary agents. If we look at the pathogenesis of viral enteritis, once a virus infects the host, these viruses generally infect cells in the intestinal villi. They cause damage to the intestinal mucosa, and this leads to several events. First, we have a malabsorption, maldigestion that's manifested as an osmotic diarrhea. So we get an increase water and electrolytes in the GI tract. We also have secondary bacterial and protozoa infections. We have immune dysfunction because we do have quite a large component of the immune system in the GI tract of poultry, and an inflammatory response. That inflammatory response can result to damages to the intestinal mucosa. So taken together, this is a situation that can cause permanent damage to the GI tract. When we think about the viruses that are most commonly associated with enteric viral diseases in poultry, we have members of the Reoviridae family, reoviruses and rotaviruses, adenoviruses, coronaviruses, and then we have a group of viruses that kind of get characterized together as small round viruses. And this is based on their appearance by electron microscopy. This is just an image that doctors Peter Woolcock and Shiva Prasad put together showing many of these viruses. And these small round viruses, well, the name is pretty descriptive. They're small, they're round, they don't necessarily have any structural features that when you see them through an electron microscope, you would know that it was a picornavirus versus a calicivirus. So very generic description of viruses here. But again, this is the list of usual suspects. We have a lot of challenges in diagnosing enteric viral diseases. It's very difficult. Historically, electron microscopy has been used to identify these viruses from fecal preparations of affected birds. Unfortunately, you need a lot of virus to be able to visualize this by EM. Somewhere on the order of 10 to the 5 to 10 to the 6 virus particles per ml is the minimum to be able to observe them by EM. We have difficulties propagating many of these viruses. Either they're very difficult to grow, or we just don't have a system that we can grow them in. So it's not possible to isolate some of these viruses. And we don't have many tools for detecting the unknowns. Advances in our molecular techniques, such as PCR and RT-PCR, where we can amplify large quantities of nucleic acid, has certainly helped us. But those assays really enable us to detect things that we already know. In order to develop those assays, we have to have the knowledge of their sequence, and so they're very useful, but they have limitations. Just because we detect a virus does not necessarily indicate that that virus is the causative agent. Many of the viruses on this list here, we can detect in chickens that are healthy. So mere detection does not implicate a virus and disease. And lastly, on the list of challenges is that it's very difficult to reproduce some of these enteric diseases experimentally. So it's difficult to prove Koch's postulates, which is what we generally expect to do when we're trying to implicate a particular pathogen as a causative agent, but very difficult to do that with some of these syndromes. I've already mentioned electron microscopy, but we can also make attempts to isolate viruses. If we're trying to isolate chicken viruses, we can try to isolate those viruses in chicken embryos or chicken cells. When it comes to the enteric viruses of turkeys, those viruses really have a preference for growing in a turkey cell. So we need to try to find turkey embryos or prepare cell cultures from turkeys. It's very difficult to find specific pathogen-free turkey embryos in the US. So that's not a readily available substrate for us to test in. Many times we find ourselves trying to isolate or culture these viruses in the host. Sometimes that's easier than working with embryos or cell culture. In addition to PCRs, we can do immunohistochemistry to try to detect antigen in tissues from these affected birds. We also have the tool of RNA or DNA probes, and we can actually perform in situ hybridization, again, in the tissues from affected animals. And we compare these results with the pathological changes that are observed in tissues. Biological characterization, again, this is an important component because, as I mentioned, many of these viruses are found in healthy animals or detected in healthy animals. So there's variability in the viruses, and they don't all cause or contribute to clinical disease. So as an example of how we utilized laboratory systems for diagnosing and identifying the etiology of running and stunning syndrome, I'd just like to share with you some of our approaches and results that we generated. So running and stunning syndrome is not a new disease. This is an enteric disease of chickens, primarily meat-type birds are broilers. It causes a decrease in body weight, an increase in our feed conversion ratio. Most of the time we see this in very young chickens, but certainly by the first or second week of life, it's visually apparent. When you walk into an affected poultry house, you'll see hatchmates that are drastically different in size. The picture that I have here is from an experimental reproduction of RSS. Both of these chicks are hatchmates. You can see the one on your left is far smaller than the one on the right. And this chick will never catch up in body weight with its hatchmate. Another common feature are thin, pale intestines with watery contents. The intestines have lost their tone You can actually see gas formation and the liquid contents of these intestines. And then finally, we have a cystic enteropathy. This is probably the most consistent histopathologic finding in RSS-affected birds. Many of these affected birds have severe cystic enteritis, and you can see these large cyst formations highlighted by these arrows. We can see in many cases an abundance of necrotic cells in the lumen of these cysts. The villi become atrophied and blunted. There's also crypt hyperplasia. And when we look at different sites of the gastrointestinal tract for the presence of these cystic lesions, we find in most cases more lesions in the duodenum compared to other locations. So certainly this virus has a predilection for that region of the gastrointestinal tract. People have been trying to determine the etiology of RSS for decades. This is, as I mentioned, not a new disease. Descriptions date back more than 30 years. There have been multiple etiologies suggested. All of the small, round, non-enveloped viruses have been implicated over the course of the past 30 years. However, when those viruses were put back into chickens as single virus inoculums, we could not reproduce the disease. This wasn't a situation where our laboratory wasn't able to do it, but many investigators have tried reproducing the disease with these single virus inoculums. Generally, we'll see weight suppression because many of these viruses do cause body weight suppression, but we don't see the body weight suppression paired with the cystic enteropathy. In addition to the small round viruses, certainly reoviruses and rotaviruses have also been implicated. So our question was, what else is in the GI tract? What else could be causing this disease? Just to give you an idea of the chronology of lesions in the field, first it's important to state that in the US, we raise our broilers on built-up litter. This is generally, these are pine shavings that are put into a poultry house, and over the course of the next year, sometimes year plus, those shavings stay in the house, and new placements of broilers are put in on top of the used shavings. Occasionally, that litter will get top dressed with clean pine shavings, but we're putting day-old chicks on this used litter. For RSS, within 4 days after placement of day of hatched chicks, we can see the first intestinal lesions forming. The peak of these growth and microscopic lesions occur between 7 and 12 days. And then after 12 days of age, we see this lymphocytic enteritis. And then from 9 to 35 days, we have the cystic enteropathy and enteritis. So one of our initial studies was to confirm that what we were seeing in the field was in fact infectious. So we harvested the gastrointestinal tract from affected broilers, and we inoculated day of hatch chicks with a filtered homogenate and with an unfiltered homogenate. And what I'm showing here, we have negative controls represented by the orange bar, and this is the average body weight at 12 days of age, 12 days post-placement. Average body weight of broilers that received the filtered intestinal homogenate and then the unfiltered homogenate. So you can see that we definitely had body weight suppression in both groups, filtered and unfiltered. And then we also collected intestines, and in both of those groups, we saw the presence of these cystic lesions in the duodenum. So from this study, we were able to conclude that we were dealing with an infectious disease, that viruses alone were capable of reproducing the disease, but that the addition of bacteria and possibly protozoa would certainly make this disease more severe. From there, we did pursue the usual virus isolation attempts where we took material from infected birds and put them into embryos and into cells. We isolated many viruses. We characterized these viruses using PCR and sequencing. And aside from finding some variant reoviruses, nothing really stood out as potential etiologic agent. So then we pursued some metagenomic studies. We infected SPF meat type chicks with our filtered intestinal material, and alongside of those chicks reared uninfected chicks, and then harvested the GI tracts. We isolated DNA and RNA from those intestines and performed a metagenomic analysis. Basically, we wanted to see if there were sequences in the RSS inoculated chicks that were not present in the uninoculated chicks. In addition to finding many sequences that were identified as unknowns, we did see a high degree of representation for Astroviridae and Picornaviridae present in the RSS infected chickens, and they were absent in our unchallenged controls. So this kind of gave us a little bit more direction. Rather than trying to look at everything, we focused our efforts on trying to determine the role of Astroviridae and Parvoviridae in RSS. We also, at around the same time, there were some publications about a chicken parvovirus from Michael Day at the U.S. Department of Agriculture, and they identified that as a possible novel enteric virus associated with RSS. We were able to amplify sequences from astroviruses from RSS-affected chickens, and one of our PhD students, Dr. Kyung-Il Kang, was able to do full-length sequencing of this astrovirus, and based on its comparison with other avian astroviruses, it was a variant, it was novel, and would be considered possibly a new member of the genus of the Avastrovirus genus in the family of Astroviridae. At this time, this was back in 2012, high throughput sequencing was still quite expensive, and so he was able to achieve obtain the full-length sequence through primer walking and amplification of the 5' and 3' ends. I think nowadays we could sequence this in a day easily, but it took him a couple of months, just to highlight some of the progress that we've made. So our question was, is novel chicken astrovirus the causative agent of RSS? And what I'm going to share with you is how we developed the diagnostic reagents that we needed to perform these studies to answer this question, what attempts or how we were able to isolate the chicken astrovirus, and then utilize this virus and the diagnostic tools to examine the pathogenesis in an experimental model, and then methods for control. So first, when you're dealing with a pathogen that people haven't really dealt with very much, you quickly realize that you don't have many diagnostic tools. Aside from the PCR, that was really the only tool that we had to use to test tissues from the field. So we felt like we really needed to generate some specific serum so that we could look at the tissues, look for staining of antigen in tissues from affected birds, and study the pathogenesis. So we were able to amplify the capsid, the gene that codes for the capsid protein for several chicken astroviruses. We have the novel chicken astrovirus, and then 2 additional chicken astroviruses called avian nephritis virus 1 and 2. And then we also amplified the chicken parvovirus capsid. These were cloned into a baculovirus expression system, and we were able to purify the protein, all 4 of these proteins, and use those proteins to immunize rabbits so that we could get a monospecific hyperimmune serum to use in other studies. These capsid proteins were also very useful because we could, and we did develop ELISAs that we used to look for antibodies specific for the astroviruses and the parvovirus. And then we were also able to use the capsid protein as a recombinant vaccine. Other diagnostic tools that we developed were aimed at looking at sites of viral replication. So we made riboprobes targeting regions of these 4 viruses, and these riboprobes enabled us to perform in situ hybridization on our formalin-fixed paraffin-embedded tissues. We had many of them from our clinical submissions. So we were able to look for replicating astroviruses and parvovirus in these tissues. In one of the studies that we did, we wanted to look at experimentally infected birds and look at the progression of or the appearance of these viruses in the GI tract. So we inoculated our day-of-hatch chicks with our RSS filtrate, and then we started collecting tissues at 6-hour intervals and looked at those tissues with our riboprobes. What we saw was an immediate presence of astroviruses. These were chicken astrovirus and the avian nephritis virus 1 and 2 in the duodenum in our RSS exposed chickens. We also saw that there were co-infections with multiple astroviruses. So what we're showing here on the top panel, this is a cross-section from our uninfected chickens. And then on the bottom panel, we have a these are the duodenal cross-sections from our RSS exposed chickens. And we can see that the chicken astrovirus probe is staining in the intestinal villi. We have avian nephritis virus 1, also chicken astrovirus staining, and to a lesser degree, avian nephritis virus 2. In our studies, we didn't detect any chicken parvovirus at any of the time points. We also used these tools to determine if any of these viruses were present in this lesion that's most commonly associated with RSS, which is that cystic formation in the crypt of Lieberkühn. And using an oral challenge model with the RSS filtrate, we did detect the chicken astrovirus here in the crypt. And this is just a close-up of that. And this was by 5 days post-inoculation. So very quickly after the chick was infected, we saw this replication of the chicken astrovirus in the site of this lesion. And that was the first time that we had a direct association between the most common histologic finding and a particular virus. And that was at 5 days post-infection. We also wanted to look at other primary lymphoid organs as well as the pancreas to determine whether or not the virus was replicating in tissues outside of the intestinal tract. As you can see here, again, we were looking at both uninfected and RSS exposed chickens. We only saw staining for these chicken astroviruses in the duodenum, not in the pancreas, the Harderian gland, thymus, or bursa. So this was an implication that these astroviruses do have a selective tropism for intestinal tissues, which was originally what we thought, but we needed to confirm that. While we were doing those studies, we were also trying to isolate the chicken astroviruses, and as I mentioned earlier, this can be a difficult process. Some of these astroviruses don't grow well or at all in our lab systems, but nonetheless we wanted to do our best to try to isolate this virus. So we homogenized the intestinal tract from RSS affected chickens. That homogenate was filtered through a 0.2 micron filter. We incubated that with Rio virus and rotavirus anasera to try to reduce the possibility that we would also isolate those two viruses, which at least Rio viruses are commonly found in the GI tract of poultry. And then we inoculated several cell lines. We have the canine kidney cell line. We have a quail fibroblast cell line, a chicken fibroblast cell line. Vero cells are primate epithelial cell line, and then an avian liver cell line. And we looked at the cells daily for any kind of morphological changes or cytopathic effects. And then when we weren't seeing cytopathic effect, we decided that we needed to look at these cells with immunofluorescence, an indirect immunofluorescence assay, because it was possible that we had virus replicating in the cells that was not causing cytopathic effect. And sure enough, in the liver cell line LMH, we did have the chicken astrovirus replicating in these cells. The bottom panel is an uninfected cell control. We did use a nuclear stain. That's what these red stains are. And then the cytoplasm of the LMH cells were staining positive for the astrovirus. These viruses do replicate in the cytoplasm. So we were able to confirm that we had isolated an astrovirus. The last thing on our list was to try to reproduce the disease. Now that we had an astrovirus isolate, we wanted to put this back into chickens and see if we could reproduce the disease. We were pretty excited at this point in time. It had taken us several years to get to this point, and we're pretty certain that this was it. We were going to inoculate these chicks. They were going to show body weight suppression, and they were going to have cystic enteropathy, and we did that, and there was no cystic enteropathy. We had some body weight suppression, but our hallmark lesion was not present in these infected chickens. So our thought was maybe we needed to back passage this and see if by passaging this isolated virus back through birds, maybe somehow we would restore the phenotype. This is our thinking, restore the phenotype of what we were seeing in the field. So we took day-old broilers, we inoculated them with the astrovirus isolates. 7 days later, we harvested the intestines. At the same time, we had negative or uninfected chickens, and we harvested those intestines as well because we didn't want to, we wanted to be able to see if there were any other components in the GI tract of these broilers that might be contributing to body weight suppression. And those intestines were processed and put into a second group of day-old broilers. And we repeated this process for 6 passages. What I'm showing here is the percent body weight suppression compared to the uninfected controls. And you can see that starting really by passage 4, we started seeing significant body weight suppression. By the 5th passage, we had a 17% body weight suppression. And then also we were looking at the duodenal loops from these birds. This is from an RSS positive control. And then pass 1, pass 3, and pass 6 from our astrovirus passages. And when we looked at the crypts from these passages, we saw an increase in the number of cystic crypts by passage, as well as the size of these cystic crypts with each passage. So this was really the first time that we or anyone was able to reproduce the clinical disease of runting and stunting syndrome. The last part of this quest for our identifying the etiology of runting and stunting that I want to share with you is the development of a capsid protein vaccine. Because we had cloned the capsid protein, we wanted to utilize that protein to vaccinate broiler breeders and then take the progeny from those broiler breeders and put them through our RSS challenge model. So we vaccinated broiler breeders 3 times during the course of the study. We brought them into production and we collected the eggs. We set the eggs, hatched the eggs, and put the progeny through our RSS challenge model. The birds, or the progeny, was not completely protected, but the capsid protein vaccine did mitigate weight suppression, and it did result in a reduction of cystic lesions in the progeny that were challenged. So it's difficult to tell how the progeny would have fared out under field or commercial conditions, because there are so many other contributing factors that the mitigation that we saw under experimental conditions may be lost under field conditions. But this was promising for us. Unfortunately, we don't have any commercial vaccines for enteric viruses. There are some companies that during this time elected to put a variety of viruses that were identified into inactivated, oil adjuvanted vaccines and use those in their breeders. When we tested progeny from those vaccinated breeders in the RSS challenge model, they weren't protected. So either the wrong viruses were included in this vaccine, or the wrong viruses were included in the vaccine. We have several other experimental vaccines that have been reported, but so far nothing that's commercially available. Just in wrapping up, there's been a great deal of advancement in understanding the virome of diseased broilers over the course of the past couple of years. As a virology community, we aren't as advanced as what I've heard over the past day as far as understanding the interactions of these viruses and the host gut, but certainly we're making some strides. NGS sequencing and metagenomic analysis has been performed and reported several times using different target populations of chickens and turkeys. These studies have been used to identify a very complex viral community in the gut. Novel viruses have been identified, unknown viral sequences, much like we found in our earlier study. And then also bacteriophages have been identified in the gut contents from diseased birds. We don't really know what the role of some of these viruses are in disease, if any. Certainly, we continue to see a high representation of our Rio viridae, Astroviridae, and Picornaviridae. What I'm showing here is just a metagenomic analysis of some commercial flocks that were having enteric issues. This was done by Michael Day at the U.S. Department of Agriculture, and he identified Astro, Rio, and picornaviruses. New viruses such as picoburnavirus, the IHSIG virus, and turdivirus have also been identified in some of these metagenomic studies, but we're still in the very early stages of understanding how these viruses might be playing a role in enteric disease. Another interesting finding from Dr. Day's study was the association of some of these enteric viruses with gut bacteria. So in this chart, the larger the circle, the more positively or negatively associated the virus is with the bacteria. The blue circles and the intensity of the blue circles indicate a very strong association between the viruses with the gut bacteria, and the red circles indicate a low degree of association. So just as an example, we have here that chicken astrovirus had a fairly high association with Clostridium and Ruminococcaceae in this study. And so this was a really interesting outcome, and I think this is more of the type of work that we'll be seeing in the very near future as we try to get a better understanding of these viruses. We need to study more the mucosal immunity in the gut-associated lymphoid tissue. GALT is the key immunologic tissue in the mucosal immune system, and a recent study that was published in the Frontiers of Immunology in May actually identified it as a command center for the humoral immune response. Of course, this work was that they reported on was actual vaccination of hens with an E. coli bacterin, so it wasn't associated with viruses, but the methods that they used to evaluate the immune response to that bacterin were perfect for what we need to do for viruses. So challenges that we have are looking at the changes in enteric viral disease, especially in the no antibiotics ever or antibiotic-free production systems. Likely these changes will also change the landscape for the viral pathogens. And certainly time of discovery. Seems like we're able to get so much more done now with all the high throughput sequencing. This will certainly generate data more rapidly than we've been able to generate in the past. So the team at Georgia that helped with the runting and stunting is Dr. Egbert Muntz. He's now with Boehringer, Kyung-Il Kang, BJ Durairaj, Eric Linneman, Vanessa, and then also a great thanks to our funding agencies.